We didn't. Does that seem familiar? Well, maybe that's the wrong question.
I think you're past that. Yeah, you went past that.
We went past this?
What's the next one?
We talked about monomeric.
Yeah.
We went through this. Okay.
That's French.
Right there.
Right here. Somebody said right here? Do I hear a second? So we did actually cover this, right? Okay. Let's see whether this thing works. Apparently it does. Okay, let's get started. Because I got a little behind in Module 1. We had to take some time for Module 1 material in Module 2 lecture time. It's put me into talk as fast as I can mode. So we'll try to get through the proteins involved in DNA replication today and hopefully start on recombination. We'll see how that works. If you have questions, please stop me just because I'm going too fast. If I'm going too slow, somebody come up here, wake me up, get me going again. I get off on tangents sometimes. So what's wrong with this picture here? We stopped here, but you get to the point where you look at textbooks and you look at figures and everywhere you see mistakes. So I was even thinking about using molecular biology of the gene at a Cold Spring Harbor, changing textbooks for next time. And I started looking at their explanations and figures about Paul 3. And I looked at it and I said, wow, this is nothing like I'm teaching my students. The 7th edition of Molecular Biology of the Gene, I had the guy come and do a sales pitch here. It's just out. Genes, the competing textbook, Genes 12 is just out. And so I'm going, which is correct? And I immediately made some slides to show you the brand new, the true picture. And then I did more reading and found that the true picture was discovered in 2011. And since then, there's been more true picture that says it's back to the old picture again. So you'll see when I get to that point. There are 2 models now for how it might be. I believe that Lewin is right. So we'll keep it like the textbook. I've become a little disillusioned at molecular biology of the gene. It looks fancy schmancy, but the grass is always greener on the other side. So I'm reconsidering changing. I think maybe Lewin is still pretty good. So what's wrong with this figure? Anybody see anything wrong with it? How about DNA B? Does that look like a big red ball? No, it looks like a green necklace, doesn't it? We all know that. It should be wrapped around one of the 2 strands, right? So we'll just say they just didn't want to put that much detail. But which of those 2 strands would it be wrapped around at the replication fork? It's a green necklace, has 6 subunits. Does it wrap around the template for the lagging strand? Or does it wrap around the template for leading strand? Remember which way it goes? It travels 5' to 3'. That means it's going to have to be encircling the template for lagging strand. I guess Lewin thought you guys would not see that. Okay, the clamp's associated with the core of the lagging strand, just associates at the end of the Okazaki fragments and reassembles the next fragment. Clamp, y'all know what the clamp is, right? Helicase DNA B is responsible for interacting with the primase G to initiate. These are just words. Right now we'll see some figures that illustrate this, but the clamp is that donut, right, that sits right behind DNA polymerase, except it's the first thing to bind, and then DNA polymerase sits in front of it, actually. So as soon as the new strand is made from the single strand template as it's chugging along, single strand on each side. The clamp is on that first part that's double-stranded, that newly minted double strand, one of the strands being brand new, still smoking hot. That's where the clamp forms, and it keeps the catalytic subunit attached to the template so it doesn't float off. Apparently, if it didn't have that beta clamp, every 10 base pairs it would lose interest and begin to float off and explore its space. But that clamp keeps it processive. Processivity is what biochemists call the property of remaining associated with the template with respect to RNA and DNA polymerase. What's this about DNA-G? DNA-G is the primer, the primase, not the primer, it makes the primer. It's a modified RNA polymerase or specialized RNA polymerase that is only active when it makes contact with DNA-B, the helicase. This keeps it from priming all sorts of places. Different enzymes are required for lagging and leading. As we know, different copies of the same monomeric unit in bacteria. It's not different subunits, it's just two copies of the monomeric catalytic domain with its own epsilon, which is proofreading, and one is dedicated for each of the strands. Of course, that general strategy of doing both strands, the strands that go with opposite polarities, which is pretty tricky, that strategy of doing them at once in the same complex is followed by all organisms, or at least non-viral organisms. More details. Let's dive into the weeds here. Replication, Paul 3. It's a giant enzyme, 900 kilodaltons. How big is RNA polymerase? About 450 to 500 kilodaltons. So this is very much larger than RNA polymerase, but it's much, much stupider, right? RNA polymerase is attracted to promoters, it's highly regulated, it stops, it pauses. Every time there's a hairpin that's formed in the RNA that comes out the back door there, it pauses and thinks about and marvels at what it just created. It goes at 50 nucleotides per second. DNA polymerase, 880 base pairs per second. Paul 3, a catalytic core. The alpha is actually the machine that makes the new strand. Dimerization component, tau, links the two cores. Processivity, beta. What does that mean? That's the beta clamp. Has two subunits, two betas. What's it called in a eukaryote? It has three subunits that make the clamp. It's called the pCNA. Okay. Clamp loader is sometimes named after its two prominent subunit families, gamma and delta. They have multiple copies of gamma, and that puts the clamp in place, loads the clamp. Here you see diagrammatic representations of some of the complexes that were isolated biochemically. My GoPro, you can talk to your GoPro and it can talk to you. It now just, it said it's saving. I guess it's got an IRA or something. It's having its own little conversation. Okay, so this is the diagrammatic view of Paul 3. These are complexes, some of which can be isolated biochemically in various sedimentation assays and purification schemes. These are natural clustering affinities that represent the holoenzyme. So you see, this thing is much more complicated than most of the other polymerases. I mean, than all of them, but DNA Paul 1, for example, that we talked about, just has one subunit, right? And then you cut it with a protease at that sensitive site, you have the clinal fragment and the small fragment. So here you see this group right here called the core. Alpha is the main event here. This is the catalytic subunit. Epsilon, of course, is proofreading. See, it's much smaller. And then you have a structural one, theta. And notice there are two of these cores that are held together by tau. Tau is this arm, this flexible arm that keeps the two tied together. Tau for tether. Because this, in a sense, it doesn't keep the DNA polymerase from floating, from disengaging from the template, but it does keep it from floating very far. So in a sense, it also contributes to processivity. It keeps these two on a leash, the hounds of hell. This is our friend, the clamp again, but now you should look at it with new eyes because you know what's there. This is the side view, right? That turns out to be part of the clamp loader. It didn't get in the structure, but this is the alpha catalytic subunit. This is the proofreading component, and this is the side view of this donut, the clamp, the sliding clamp. And if you flip this thing over and look on the bottom, you'll see the clamp is a donut, 2 beta subunits, and DNA fits right in the middle. Okay, let's further construct the replicase. We've got the 2 monomeric units here. We've got them connected by tau, and this is what they're connected to, the clamp loader. We're going to see some other representation. This is just little balls, but you'll see we have some more structural information where we can make crude approximate cartoons of how it all works, and we'll see that in just a minute. But there's only 1 clamp loader per replicase. This has to load the clamp wherever it is needed. It's only needed, though, usually on the template for lagging strand, right? Because lagging strand is reinitiated over and over and over again, so clamp loader is largely devoted to putting the clamp behind the polymerase, except it puts the clamp first and then polymerase is loaded in front of the clamp. Sometimes I'll ask a question like, which of the following subunits is found only once in Paul 3? Well, you know now what to look for. Everything is twice except components of the gamma delta, which is another name, components of the clamp loader. There's only 1 clamp loader, but 2 of everything from here up. So the last part, of course, to make the holoenzyme is addition of the clamp itself. So all of this together is considered the holoenzyme that forms the replicase, DNA Paul 3. So how many, what gene encodes Paul 3? Lots of genes encode Paul 3, each of the subunits. Now here's sort of a party question that you'll be the belle of the ball. If you know the answer to this, you can kind of mosey up to someone you'd like to meet. You're kind of sloshing your Diet Coke with the ice, and you're going, by the way, which of the subunits, you don't mind if I butt in, do you? Which of the and they're like, who is this guy? And you take a little sip very slowly, because you're in command. You go, by the way, do you know which of the 2 subunits of the replicase are encoded by the same gene? Oh, it's quite mysterious. It's the DNA X gene. And they go, nah, really? Get out of here. No, really, it is. There's a gene that encodes both of them simultaneously. It's just a shift in the reading frame. That's a viral trick. Hoof and mouth disease uses that kind of trickery to cause the ribosome to shift reading frames. E. coli's been doing that for years. There's a 7-base nucleotide sequence. It's all A's and the last is a G. If you have that, it's a configuration that confuses the ribosome. It'll skip the reading frame, get out of line. These 2 genes, gamma and tau, have the same gene, but different reading frames. Gamma and tau. Be daggum. See where that gets you. But the night is still young. Gamma and tau. Man, isn't that something? Wouldn't that impress you? Okay. Assembly stages. Here you see the clamp loader, and here you see the clamp getting ready to load. Now, what does the clamp load onto? It's looking for duplex nucleic acid. The first thing to be made in the lagging strand is the primer, right? And then the polymerase turns that into DNA, extends it as DNA. So the clamp actually first binds around that new primer and the template. So it's really binding at first around a DNA-RNA hybrid. It has the potential to bind around a DNA duplex. But what actually happens in replication is the clamp is assembled around the primer. And when it arrives on the scene, it bumps or displaces the primase itself, DNA G, knocks it out of the way, sits down, clamps down, and now is waiting for Mama Duck herself. DNA Pol 3, the catalytic subunit, alpha and epsilon and theta, they come and they sit right down, make all the connections, everything's locked in, and then they go. Here you see it is now clamped around the RNA primer and the template strand, and here you see it being joined by the core, one of the cores of DNA polymerase, being that red goose egg there. The clamp, of course, is the primary contributor to processivity. So it's a little bit of contribution by tau. You don't see tau here. Where would tau be in this diagram? Can you remember? Tau, two copies of tau are coming out the head. It's like a two-headed monster. This is the body. And tau here, tau there. And at each of the ends of these stalks, you're going to have the core. One copy of core, another copy of the catalytic core. Here you see the two-headed monster. The body, the clamp loader, tau, one monomeric unit, the other monomeric unit. Well, now you guys already know that. Here is, apparently I have this fascination with the clamp. Here's yet another picture of the clamp. How many helices does it have? If you count them, you'll see there are 12. We have 12 also, except our sliding clamp. This is where DNA goes, right in the middle. Our sliding clamp is split up into 3 subunits, the pCNA, but it's still 12 helices. Here you see the pCNA, each a different color. It's the same, very similar structure. The beta clamp dimer is ring shaped. Assembly or removal requires energy. So it doesn't take any energy to fasten the clamp around the duplex. It just takes energy for the clamp loader to disengage itself because it just loves what it just did. It went, whoa, that is so cool. ATP is then triggered to be burned, and it goes, it loses interest and it is locked into an inactive ground state, waiting for its next turn. We'll see more about that in a minute. And this is what you can do with an iPad Pro. You can waste your whole weekend making little silly graffiti. This is the clamp loader. GoPro. Take a nap. This is from the molecular biology genes book, just redone, traced, and drawn a little bit myself. In the molecular biology of the gene book, they had this part wrong, so I redrew that. They had it bound to a fully formed duplex when it really only binds to the primer, which is represented in red here. But notice the role of ATP, because this is a common theme in molecular machines. It's called an enzyme, but this enzyme doesn't catalyze the breaking and forming of bonds, of chemical bonds. It does work like a little machine, like a little robot. And ATP is used to lock it into various conformations and do work that way. So let's look, and we'll see this in 3 or 4 different machines that we will cover. And let's see how this works. You have the clamp loader. It's in a ground state, unable to bind. You'll notice the clamp is going to go on the bottom here. The tall subunits are up here. We're just looking at the clamp loader. It's in this inactive form. It binds ATP. It doesn't burn ATP. Just the binding of ATP locks it into an altered state where now it is receptive to binding its 2 substrates. What are its 2 substrates? The clamp itself and single-strand DNA at the replication fork. Let's see how this works. Notice how it's opened up here, and now you have the clamp, and it can now bind the clamp, and it binds the clamp in such a way as the clamp can't really fit unless one end of the 2 subunits opens up. So now it's in this configuration, which is just perfect for loading a duplex. And once it gets in this configuration, it's attracted to this single-strand DNA region that's at the replication fork. And when it does that, it automatically loads the DNA duplex region in the interior on the inside of this clamp with a break. Once it gets in this configuration, so it has affinity for the clamp, which it opens, and then it has affinity for the single-strand DNA at the replication fork. And when it binds the DNA, after binding the clamp, second DNA, that act triggers the hydrolysis of ATP. So now it becomes ADP. Well, didn't correct that correctly. That needs to be an ADP plus pyrophosphate. I'll make that correction in the slide. And when that happens, the ADP is released and it snaps back into that original ground state, waiting once again to interact with ATP. That's a machine that conformation is controlled by the binding and hydrolysis and release of ATP. We'll see several, and I'll point these out to you when we get there, but this is a common way molecular machines work. They use ATP to lock in a configuration. So it doesn't go all the way back to this state. It means it releases it instead of removing the DNA, opening the ring, and then releasing the ring. It's locked in here, so it's off, and you're stuck with the clamp loaded. So this is a little crude thing that I did 10 years ago now, but it very inefficiently tries to instruct about how all this works. So bear with me. And see if you can sort of talk your way through it also. Because when you're studying, you need to understand the things, the concepts completely. Can you talk yourself through DNA replication with Paul 3? So let's look at the components. Gamma is the shorthand for gamma delta clamp loader. It has 2 tall arms coming out from it, and they're ultimately attached to these 2 kind of blue things, Paul 3 catalytic subunit. And notice there's also a connection with the helicase. The tall has a connection with the helicase, and it has a connection with the catalytic components, the monomeric units of Paul 3. This connection here is what pulls the rest of it along. It would move on its own, just like RNA polymerase moves on its own as it makes RNA, base by base. But this helicase pulls it along so it goes 10 times faster than it normally would if it wasn't connected there. Pulling it along, and it's pulling these two, and it's grabbing it by the taw. Notice you have a clamp down here and a clamp there, and you have one sitting on the clamp loader. So let's see how this progresses. In this configuration, you have this open stretch of template for lagging strand. This is lagging strand. It's going the opposite direction as the replication fork itself. This is leading strand up here. This catalytic subunit and its clamp remain attached until it hits a lesion of some sort, a thymidine dimer, and sometimes it doesn't hit anything. It'll stay there the entire, all the way halfway around the genome till it gets past the tetracytes. But this one's constantly, every 1 to 2,000 bases, reinitiating because you keep getting this exposed region of template here. Now, what's not shown here is the binding of what? Single-strand binding protein. That is always going to be there because this is a considerable stretch compared to how much you see here. And DNA G is always looking for single-strand DNA and an opportunity to come in here and make a primer. It's only active if it can make contact, though, with the helicase DNA B. So there it goes, it makes contact, and now it's activated, now it's looking for its particular little sequence. And I can't remember the name of it. It's very much like the DAM methylation site. It's GCTA or something like that, which it finds and then actually begins to prime at that point. And here you see it's going to make a little primer, hands-free. Look at that, right underneath, like it laid an egg, it's making that primer. And as soon as it makes that primer and it begins to peek out like a new baby chicken, why, clamp loader goes, all right, I'm on it. It is actually displaced by the clamp swinging around, the clamp loader swinging around and locking a new clamp on the RNA DNA duplex there, displacing DNA G. Well, notice what's happening down here. Down here, the polymerase is beginning to finish up. It's beginning to bump against the previous Okazaki fragment, which of course has a piece of RNA as primer. They all start with the primer, and here you see the new primer being laid down there. When it hits here, it collides and it releases. And now it is ready to be slammed down here onto that clamp that's preformed to start again. And this clamp is not shown here, but this clamp falls off when it's not very stable when it's not in contact with the DNA polymerase. And here you see the new strand. The clamp is loading again, and so forth. So you should be able to kind of talk your way through all the steps. This is some from various review articles. You get a little better picture of it. The clamp here is red. Here you see the clamp loader, the two TAWs, and here you see DNA polymerase, one of the catalytic subunits. Here you see the other catalytic subunit. Each has a clamp behind it. The primase is there. Notice the little primer that's made, the RNA primer. Notice they drew the helicase on the correct strand, the template for lagging strand. And the rest of it, you can see how this now swings up, bumps off the primase, and puts that clamp there. The helicase increases replication rate by 10-fold, and it activates the primase. The contact between the DNA G protein, the primase, and DNA B is required before you get the primer made. Here you see it, what happens as it bypasses damage. There's the lesion. That could be most anything, a thymine dimer. But you see it disengages, the polymerase comes loose. Here's the lesion down here, the old clamps down here, and it rejoins up here. You don't need to know this mechanism, because I'm not sure that it really works like this, because you have to have the primosome activating in here someplace. So this is kind of a placeholder to know that people have been thinking and making models about how the replosome negotiates damage, how it has to reinitiate again. So you're going to have to reinitiate the leading strand. But what we see right here is a movie that should change the way you think about life itself. This is to convince you that it's just impossible. You have molecules in the dark just doing their thing. That's all they know how to do. And they're making DNA with high fidelity at 880 base pairs every second. That includes time to try out the wrong base and then get the right one and then back up and all that stuff. It's happening in the dark, no brain. It's just like salt crystallization. We've isolated it. It can happen in a test tube. It's just chemicals. The Nobel Prize was given for it, but they were looking at Paul 1 at the time. This is pretty amazing. And this is a fairly accurate representation of what happens now. All that buildup, I hope this link works. Can you recognize the parts? The helicase. The helicase. The alpha complex for leading strand. And look how the clamp being loaded, it coming off and going right back where the clamp is. Clamp being loaded. DNA replication. It has sound, but I don't know how to activate it. Usually it's activated automatically. I don't know how to work the podium. Can you identify the parts? That is happening in you. It's happening in your cat. It's happening in bacteria in your gut. It's a fundamental process of all of life. Who directs it? It can't help itself. It does it in the dark, day and night. A crash course? Oh, cool. She doesn't have any sound either. Okay. Well, look, another replication movie. Unexpected error has occurred. Okay, that's just to make me look bad. Okay, so we'll keep moving. You've seen the major event here. That movie is pretty cool, and you have the link there. I recommend just staring at that like all Saturday morning. See how it changes your life. Okay, this is the figure. Then, of course, I took it in my iPad Pro and messed it up. But this is the figure that I scanned from the molecular biology of the gene. This three-headed monster here, that's what those people who have this textbook think it looks like. Three, there are only two strands. What's going on? Well, it looks like two of the catalytic subunits are engaged in taking care of the lagging strand. One is just finishing up as it begins to bump into the 5' end of the previous Okazaki fragment. And the other one is in the process of loading the clamp, and the polymerase now is joining it. Why did they have this? Why are people confused about this? Well, it's a long story, but part of it is the role of gamma. You remember tau and gamma? You notice how this has too many taus, This actually is three-headed beast here, has no gamma. That was part of the model. Yet gamma is highly conserved, and that frame reading thing happens with about equal frequency as the synthesis of tau from DNA X. You can actually grow gamma deficient mutants. They can fix the frame shift, so it just makes tau. It's still a viable bacteria, but it has increased UV sensitivity for some reason. It looks like gamma really is a part of the clamp loader, and that there are only 2 taus attached to catalytic subunits. It may not be this way. These are models. They're still in dispute. But I'm beginning, after looking at this and getting all excited, I think really our old friend, the two-headed beast, one for each strand, is probably the way it is. It seems logical, but you know, I have the mind of a tailless primate. I don't know. But anyway, these are some of the things that are still, I mean, something as basic as this is still in dispute. It's thought that eukaryotic DNA polymerases, replicases, just have 2. So you make the least waves in our understanding of things if you say they're just 2 heads. We don't need 3. But I thought it was interesting that it goes back onto this unusual gene, DNA X, that makes both of the disputed members here in these 2 contrasting models, gamma and tau. Okay, so that's what the replication machinery looks like. The holoenzyme, it's the most complicated of the DNA polymerases. It has the fewest molecules per cell. Let's go to finish up with some of the processes required. Each Okazaki fragment, of course, starts with a primer that you have to remove. I just lost my laser pointer. The people at home will love that because now I have to point with the mouse and they can finally participate. So you have to do 2 things. You have to remove that primer, which is RNA, and secondly, you have to join the ends together with a ligation reaction, which is an entirely separate enzyme. So what does the removal of the RNA? DNA Pol 1, right? Full-length DNA Pol 1 because it has Nick translation activity. It can bind at the nick between the 2 strands, between the 2 strands, and chew away the RNA primer in front of it using its 5' to 3' hexanuclease activity. Once it removes the primer by Nick translation, then it is that nick. You know, a nick is not the same as a gap, right? A gap would actually have the nucleotide missing. A nick is just a break in the phosphodiester backbone. That nick now is sealed with DNA ligase. What is the structure of the eukaryotic replication fork? Well, here you see Lewin is really full of, he's on a full roll, pumping out these figures. He always gets confused with the helicase, doesn't he? Here he's finally drawn some subunits into the helicase, but does he have that thing binding to double-stranded DNA? He's got to know better than that, right? It doesn't bind double-stranded DNA, it binds to single-strand DNA, but does it bind over here or right over here? Remember, this is a eukaryote. The polarity of the helicase, what is the helicase in a eukaryote? MCM2 through 7. That thing goes in opposite polarity. It goes 3' to 5', so it's not going to be around the template for lagging strand. It's going to be around the template for leading strand in a chicken or a sparrowhawk or an eagle. There are 2 polymerases that are devoted to synthesis. One for each strand, and these are specific. It's not two copies of the same one. They're two separate ones. So there's delta and epsilon. Epsilon for leading strand. Lead, E, epsilon. Delta for D, for duh, for lagging. Delta, the laggard strand. E for leading. Boy, I just gave you 2.5 points right there. I won't get any thanks. Okay. What is DNA G? What's the primase in a eukaryote? It's called polymerase alpha, and this is really two polymerases into one enzyme. Each subunit has a different catalytic activity. It has an RNA polymerase in the same complex as a subunit with a DNA polymerase. And of course, it makes the primer, which is a chimeric molecule between RNA and DNA. Remember the name? It's iDNA. So you'll see here that the DNA Pol alpha is drawn as one, I guess, yellow ball here. But actually, it has two polymerases inside of it. So it says on the slide 3 different DNA polymerases make up eukaryotic replication fork. That's true. It would be the DNA polymerase in Pol epsilon, the DNA polymerase in Pol delta, and the DNA polymerase in Pol alpha. Of course, there's an additional polymerase in alpha, and it's an RNA polymerase to make the primer. So it's drawn here with Pol alpha, priming the lagging strand, and that's what it does mostly because that's the one that needs all the priming, but it also switches around and primes the leading strand also. Okay, the next slide is in E. coli. How does the replosome handle a lesion when it runs up to DNA damage like a thymidine dimer? There's an intricate series of events that leads to termination of Pol 3 and some repair going on, and then when you're ready to go again, you actually initiate with a totally different enzyme. Let's see how that works. So here you see just sort of a general outline of what happens when the replicase runs up against a damaged region. The damage here is represented on that upper strand as these 2 black bases. That could be a thymidine dimer, for example. You see the enzyme there. It stalls because it can't put bases, it can't complement bases to a thymidine dimer because they don't fit in the active site correctly. So the replosome stalls, it comes off, and it's replaced by a repair operation. And then after the repair is over, you have to reprime. And so what you see here in that next to the bottom one is the primosome doing the priming, and after priming is complete, then you start off with DNA polymerase again. Let's see some more details in the next slide. Here on this slide, you see up the top normal DNA synthesis. The DNA polymerase is advancing along, leading lagging strand, and then at the bottom you see 2 kinds of damage. The damaged base like a thymidine dimer, as we've discussed before, the third one down. But look at the last one. What happens if you have a nick, just break the phosphodiester backbone in one of the strands? Here you see a nick in the template for the leading strand, but notice that really ends up being the same as a double strand break because there's nothing now to keep that connected because you haven't really made the second strand. So you really have a pretty severe defect. Even by a single nick in one strand, it can lead to real problems. This sort of sets the stage for a recombination/repair event involving Rec-A, which is sort of a good segue into recombination. I mean, recombination has sort of a esoteric benefit, right? It belongs to the generations in the future. But it's heavily selected for and it's in everything. How did it arise if it's so widespread and still around? Probably it arose from DNA repair. Because DNA repair is of much more immediate need and consequence. So what you need to know at this level before we get into the real nitty-gritty is which of the polymerases are devoted to this repair bypass type of reaction when you have a lesion, and that would be 4 and 5. They are both error-prone, the rest are high-fidelity. Replication stall, damage excised. Retrieval repair, this is kind of a neat little animation. Retrieval repair, and this involves a recombination-like event. Rec A is involved here. You'll have the new strand just made, that's the original template, but notice the original template for leading strand has damage. It's not clear what should go there. What happens is the new strand that's complementary to the old strand comes to the rescue and moves from here to there involving Rec A and now fills that gap. Well, look, there it goes. And then Paul 4 and 5 or 5 resume. Primosome is required to reinitiate the leading strand. So this is a chart that compares eukaryotes and bacteria, E. coli being the example. You should be familiar with a lot of these things. You already are by now. Let's go back here. You can see helicase, DNA B, MCM. Down here, another one you should know. DNA G is Paul A. The clamp is PCNA. The clamp loader, here's a new one. The clamp loader, the gamma delta in bacteria is the RFC, replicating factor C in eukaryotes. You have the dimeric, 2 copies of the monomeric unit for Paul 3 in bacteria. In eukaryotes, you have distinct enzymes, epsilon and delta, devoted to the 2 strands. And then down here at the bottom, here's a difference. Paul 1, by Nick translation, removes the RNA primer in lagging strand. In eukaryotes, there is no 5' to 3' Nick translation on any enzymes. Even in bacteria, it's only on one enzyme, DNA Paul 1. So a slightly different mechanism is in place involving an exonuclease called Fin1. In eukaryotes, there's no polymerase that has 5' to 3' Nick translation activity. How do you remove the primer? And here you see this Okazaki fragment with the RNA primer. You see it being bumped into by the new Okazaki fragment. Notice it's displacing the 5' end, and the RNA primer is waving around in the wind. You know, displacement loops. You can easily get a polymerase displace a strand that's in front of it. So this now becomes a substrate for Fin1. It comes and just cuts the primer out. It recognizes structure, which is displaced flap, and that's the end of at least the DNA replication part, and we'll start on recombination next time.